Humoral immunity. B cells develop from stem cells. So normally in adults, it develops in the red bone marrow. In the fetus, it develops in the liver. B cells mature in the bone marrow. Once mature B cells migrate to the lymphoid organs. So they travel to the lymph tissue and the lymph. B cells are not activated until recognition of specific antigens. So a mature B cell has to recognize epitopes. So this is called clonal selection. So there are two types of antigens that B cells would recognize. The T-dependent antigens, in which the antigen is presented with the self-MHC. MHC stands for major histocompatibility complex, that is expressed on mammalian cells to the T helper cell. The T helper cell then produces cytokines that activate the B cell. The second type of antigen is called the T-independent antigens, and that stimulates the B cell to make antibodies. So here's a picture of some antigens that we have present. So we have a square and we have a circle. Which one is specific for this B cell? So we see that the square is specific for this B cell. So that is going to activate. So before this gets turned on, it has to recognize that particular antigen or that particular epitope. Once we do that, we're going to go ahead and make more of these particular type of B cells, and then that's going to lead to the production of antibodies. So let's take a closer look at that. Clonal selection. In clonal selection, the B cell surface receptor recognizes the antigen. The B cells then go through proliferation, in which you're going to have the same immunological specificity. The B cells are going to differentiate into memory cells for long-term immunity and into plasma cells, which are antibody producing. So it's going to produce these antibody producing plasma cells, which are going to secrete antibodies at about 2,000 per second. Why don't we attack our own cells? Well, we don't attack our own cells because the body doesn't make antibody against itself. This is called self-tolerance. The body's not going to make antibodies against its self-antigens. There's something called clonal deletion in the embryo that occurs in the thymus. During fetal development, any T or B cells that will respond to antigens present in the body are destroyed before they could do damage to your tissues. So we damage any T or B cells that will go after our self-antigens. Do you think there's a chance that this may not work sometimes? Yes, you are correct. We see this in type 1 diabetes. So let's take our clonal selection, and we know that the B cells mature in the red bone marrow. From there, they then travel to the lymphoid tissue. Here, they're going to be challenged by antigens. And again, we have 4 different B cells here, and one type of antigen, which is like a diamond shape or square shaped, if you want to call it. Okay, which one of these antibodies is going to be specific for this antigen? Right, it would be antibody number 3. So antibody number 3 will have that diamond shaped structure here that will allow it to bind to this antigen. So are we going to make more of 1, 2, or 4? No, we're going to make more of number 3. Remember, we're going to differentiate into memory cells and into plasma cells. The memory cells are for long-term immunity. The plasma cells are then going to continue to be proliferated and go on and make more. And then we're going to see that we create plasma cells, and then those plasma cells then secrete the antibodies at about 2,000 per second. And then the antibodies are going to go on. So what happens once we produce those antibodies? Well, one, we can have what they call the enhancing of phagocytosis. First step is agglutination with IgG and IgM. These are going to cause the aggregation of antigens for more efficient phagocytosis. So instead of the macrophage coming along and engulfing just one of these bacteria, we could bind the two at a time and aggregate everybody in one area so that when the phagocyte comes along, it could eat up everything. Second way to enhance phagocytosis is something called opsonization. Opsonization is the stimulation of phagocytosis. So you're going to put antibodies all around this bacteria. And now the phagocyte is going to come along and be like, oh, I see it. It doesn't even have to recognize that that's something foreign. And it's going to eat it up. Another antibody activity is called the antibody-dependent cell-mediated cytotoxicity, ADCC. In this process, the antibodies coat an infecting cell, usually a large parasite. So here's a large parasite, and here are the antibodies coating them with their Fc stem region facing out. The NK cells, which are your natural killer cells that have lysing ability, your macrophages, your neutrophils, and your eosinophils have receptors for the Fc region of the antibody. So a macrophage, here it is, here's your eosinophils, are going to be able to bind to the Fc region. From there, once they bind, they cause the secretion of lytic enzymes. So here's the secretion of the lytic enzymes to destroy the parasite. And then, of course, the parasite gets broken up into smaller pieces, and hence we get rid of that parasite. Another function or antibody activity is neutralization. Here we see IgG antibodies blocking the attachment or blocking the active sites or binding sites of toxins. So normally a virus or bacteria or toxin would bind to something, but antibodies are going to bind to these antibodies, are going to bind to the toxins or to the bacteria or to the virus so that they cannot bind. And then finally, you can have activation of complement for lysis, inflammation, and opsonization. So let's take a closer look at the complement system. But before we do that, let's wrap it up. So here is all of those antibody activities put together. We're going to cover the complement system, and that was actually covered in Chapter 9. So you might have to go back to Chapter 9 to read about the complement system. But the reason why it's in Chapter 9 is because the complement system is actually a non-specific response. But this system works along with the normal functioning of the antibodies, which antibodies is a specific response. Okay? So the non-specific response, meaning that it assists the antibodies. Okay? There are 3 functions to the complement system. It can cause lysis, inflammation, or phagocytosis. Let's start with the inflammation first. Inflammation, meaning that it can enhance the release of histamine by other cells, which increases blood flow into the injured area. We know that we have phagocytosis in terms of opsonization. Opsonization is the coating of antibodies to make the target easier to engulf, so it attracts phagocytes. And then let's finally take a look at the lysis. Lysis involves something called MAC. MAC is a membrane attack complex. 5 of the complement proteins interact to form a unit called the MAC. This forms pores and destroys the target cell by allowing fluid to flow into the cell, causing it to burst. This is also known as cytolysis. The complement system works with 2 different pathways. The antigen-antibody reaction pathway, which is the classical, and the proteins B, D, and P, and a pathogen, and that is your alternative pathway. The serum proteins are then activated in a cascade. Okay? The components that are activated in the classical pathway are C1 through C9 proteins, and in an alternative pathway, it's C3, B, D, P, and a pathogen. Let's take a look at the first pathway. The first pathway is called the classical pathway, and this is the more rapid pathway that activates the complement system. It begins when C1, here's C1, attaches to an antibody that is already attached to an antigen, so a bacterial cell wall in this example. The complement protein acts as an enzyme, catalyzing a series of other reactions involving other complement proteins. This ends when C3 is formed. So C3 is an inactive form, and C3 is then converted to C3A and C3B. C3B binds to the surface of an antigen and causes opsonization and cytolysis. C3A leads to inflammation. The alternative pathway is the second pathway. It is less effective and slower, but it works without the presence of antibodies. The three different complement proteins, B, D, and P, act together when they're triggered by a foreign material. So here you see that they're binding to another cell wall of a bacterium. Here we'll see that the B, D, and P stands for properdin. Factor P stands for properdin, factor B and D. So that's why it's also sometimes called the properdin pathway. These guys bind to C3. This leads to C3, which is the inactive form, and the end result is C3 to C3B and then C3A. And of course, that will lead to opsonization, cytolysis, and inflammation. And here it ties it all together for you, showing you the inflammation of C3A. We can see opsonization with the C3B, and then here we can see our 5 complement proteins coming together and forming a pore and causing cytolysis. Here's a picture of cytolysis occurring to a bacterial cell here. Going a little further with C3A in terms of inflammation. C3A and C5A bind to mast cells, basophils, and platelets and triggers histamine release. This increases the permeability of the blood vessel and it increases immigration. C5A can also be a very strong chemotactic factor, so it calls these other leukocytes to come and help fight. Some bacteria can evade the complement system, so capsules can prevent complement activation. You can have surface and lipid carbohydrate complexes that prevent the formation of the membrane attack complex, or MAC. And you can also have the enzymatic digestion of C5A. So we just talked about the plasma cells. Now let's talk about the memory cells, how memory gets involved. Antibody titer is the amount of antibody in the serum. It's the level of antibody activity. Okay? Now, the first response is called your primary response. The primary response is your initial response to antigen exposure. Now, there is a delay since the antigen must activate the B cells to produce the antibodies in the first place. So when we see the antigen, right, remember we have to first recognize the antigen. Then, after we recognize the antigen, then we have to make more of those same B cells that recognize the antigen. Then we have to differentiate into the memory cells and into the plasma cells. Then the plasma cells have to make what? The antibody. So that's why we see a slow rise in the antibody titer. So usually our antibody titer doesn't peak until approximately 2 weeks after exposure. Once the antigen is gone, then the antibody levels could then begin to decline. There are 2 types of antibodies involved in the primary response, IgM and IgG. IgM is the first to appear in the bloodstream. is produced by the B cells that don't make any memory cells so that they can work more quickly. IgG are cells that produce memory cells as well. So IgG is usually the second one to come. So IgM comes first, then comes IgG. Your secondary response is brought about by the activity of your memory B cells. The memory cells are going to rise to higher levels and more rapidly. So the memory cells are going to change the plasma cells for a more intense response. So your antibody titers rise to much higher levels than what they were before and much more faster, more rapidly. Remember, memory cells can survive 20 plus years, so years or decades later. This is the reason that immunization works. Now in the primary response, IgM was the first to appear, then IgG. But at this time, IgG is going to peak first and it's going to have the most. So in this picture we have our antibody titer over the number of days. This was the first time that you recognized the antigen. So your body has to go through all those processes discussed before. Recognize it, make more B cells, differentiate into memory cells, plasma cells, and the plasma cells have to make the antibodies. So who's the first to appear that we see? IgM. Then comes IgG. And notice I said it doesn't peak until approximately 2 weeks. So here we are at day 14. And then of course once the antigen is gone, then we see a decline in the antibodies. However, if we meet up with that same antigen a second time, this time who's going to peak first? IgG, because it made the memory cells, and it's going to peak faster and much higher. So we're going to have a more rapid response. But notice IgM's response. This is the same normal response as normal. Acquired immunity. Acquired immunity is developed during an individual's lifetime. So it develops to a specific antigen after being exposed to it at least once. We're going to take acquired immunity and break it into two parts: active immunity and passive immunity. Active immunity is produced by antibodies that develop in response to antigens. So it's an actual immune response. Passive immunity is produced by transferring antibodies from one individual to another. So if we took a look at naturally acquired immunity over here, and we started with naturally acquired active immunity first, we'll see that that develops after exposure to antigens in the environment resulting from infection. So the antigens was on the door handle and you touched it and then you went and ate something and you put your dirty fingers in your mouth. The antigen enters your body and causes you to perform an immune response. However, with naturally acquired passive immunity, mother's antibodies can be transferred to the offspring through the placenta, which is transplacenta, or through breast milk via the colostrum. Did the baby make the antibodies? No. The baby didn't make the antibodies. They was passed on to the baby from the mother. Artificially acquired immunity develops after injection of the antigen. So usually we see that with vaccination. Okay, so here we'll see that artificially acquired, you're given the antigen through the vaccination and your body is going to respond to it. Artificially acquired passive immunity is injection of an antibody into a person to fight off the infection when they have been definitely exposed. So someone that's been exposed to rabies, got bit by a dog, they're given rabies shot when they go to the emergency room. That is an example of passive. Is their body creating the antibodies against the rabies? No, they're not. So hence, they are going to be producing, not producing antibodies they're given. So that's why they have to go back for series of shots. Here ends part B of chapter 10.